Hello, students. Welcome to the fourth session of Principles of Inheritance and Variation. The mechanism of sex determination has always been a puzzle before the geneticists. The initial clue about the genetic or chromosomal mechanism of sex determination can be traced back to some of the experiments carried out in insects. In fact, the cytological observations made in a number of insects led to the development of the concept of genetic or chromosomal basis of sex determination. Henkin in 1891 could trace a specific nuclear structure all through spermatogenesis in a few insects, and it was also observed by him that 50% of the sperm received the structure after spermatogenesis, whereas the other 50% sperm did not receive it. Henkin gave a name to the structure as the X body but could not explain its significance. Further investigations by other scientists led to the conclusion that X body of Henkin was in fact a chromosome, and that is why it was named the X chromosome. It was also observed that in a large number of insects, the mechanism of cell determination is of the X0 type, that is, all eggs bear an additional X chromosome besides the other chromosomes. On the other hand, some of the sperms bear X chromosomes, whereas the others do not. Eggs fertilized by sperms having X chromosome become females, and those fertilized by sperms that do not have an X chromosome becomes male. Due to the involvement of the X chromosome in the determination of sex, it was designated to be the sex chromosome, and the rest of the chromosomes were named as autosomes. Grasshopper is an example of X0 type of sex determination in which the males have only one X chromosome besides the autosomes, whereas the females have a pair of X chromosomes. So the male grasshoppers have 23 chromosomes while female grasshoppers have 24 chromosomes. These observations led to the investigation of a number of species to understand the mechanism of sex determination. In a number of other insects and mammals, including man, XY type of sex determination is seen where both male and female have the same number of chromosomes. Among the males, an X chromosome is present, but its counterpart is distinctly smaller and called the Y chromosome. Females, however, have a pair of X chromosomes. Both males and females bear same number of autosomes, hence the males have autosomes plus XY chromosome, while females have autosomes plus XX chromosomes. In human beings and in Drosophila, the males have one X chromosome and one Y chromosome, whereas females have a pair of X chromosomes besides autosomes. Let's look into the sex determination mechanism. They are of two types. The first type is male heterogamety, where males produce different types of gametes in terms of the sex chromosomes. They are of two types, XXXY type and XXX0 type. XXXY type can be seen in Drosophila and human beings, while XXX0 type is seen in grasshopper. The next sex determination mechanism is female heterogamety, where females produce different types of gametes in terms of the sex chromosomes. They are of two types, ZZZW type, ZZZ0 type. ZZZW type is seen in fowls and most of the butterflies, while ZZZ0 type is seen in moths and some of the butterflies. You have already learned about the sex determination in humans. Let's have a recap of that. Out of the 23 pairs of chromosomes present, 22 pairs are exactly same in both males and females. These are the autosomes. A pair of X chromosomes are present in the females, whereas the presence of an X and Y chromosome are determinant of the male characteristic. During spermatogenesis among males, two types of gametes are produced. 50% of the total sperm produced carry X chromosome, and the rest, the 50%, has Y chromosome besides the autosomes. Females, however, produce only one type of ovum with an X chromosome. There is an equal probability of fertilization of the ovum with the sperm carrying either X or Y chromosome. In case the ovum fertilizes with a sperm carrying X chromosome, the zygote develops into a female, and fertilization of ovum with Y chromosome carrying sperm results into a male offspring. Thus, it is evident that it is the genetic makeup of the sperm that determines the sex of the child. It is also evident that in each pregnancy, there is always 50% probability of either a male or a female child. It is unfortunate that in our society, women are blamed for producing female children and have been criticized and ill treated because of this false notion. Female heterogamety can be seen in the sex determination of birds. In order to understand female heterogamety, we name the sex chromosome differently. X is replaced by Z and Y is replaced by W. So ZZ is male and ZW is female. Females produce two different types of gametes, one with Z chromosome and the other one with W chromosome. Males produce only one type of gamete, that is with Z chromosome. The egg or ovum is responsible for the sex of the chick. Let's look into the sex determination of honeybees. The sex determination in honeybee is based on the number of sets of chromosomes an individual receives. The queen lays two types of eggs, fertilized and unfertilized. Fertilized eggs have 32 chromosomes, while unfertilized eggs have only 16 chromosomes. Unfertilized eggs develop parthenogenetically into males and are called drones. If the fertilized eggs are fed with royal jelly, they will develop into queen. If the fertilized eggs are fed with bee bread, they will develop into females which are sterile and they are called workers. This type of sex determination is called haplodiploid sex determination. Males produce sperms by mitosis, so they neither have fathers or sons, have a grandfather, and can have grandsons. Mutation is a phenomenon which results in alteration of DNA sequences and consequently results in changes in the genotype and the phenotype of an organism. In addition to recombination, mutation is another phenomenon that leads to variation in DNA. One DNA helix runs continuously from one end to the other in each chromatid in a highly supercoiled form. Therefore, loss or gain of a segment of DNA result in alteration in chromosomes. Since genes are known to be located on chromosomes, alteration in chromosomes results in abnormalities or aberrations. Chromosomal aberrations are commonly observed in cancer cells. In addition to the above, mutation also arise due to changes in single base pair of DNA. This is known as mutation. A classical example of such a mutation is sickle cell anemia. There are many chemical and physical factors that induce mutation. These are referred to as mutagens. UV radiations can cause mutations in organisms, so it is a mutagen. In this session, you have learned about sex determination and mutation. With this, we come to the end of today's session. Thank you and have a nice day.